Hello everyone, welcome to Decode Microbes, and in this video we are going to quickly discuss about the cultivation of virus. So let's get started. Viruses are obligate intracellular parasites, and unlike bacteria which can be grown on artificial medias, viruses cannot be grown like that since they require living cells or tissues for their multiplication. And when we say cultivation of virus, we mean by placing the virus in a suitable living system so that the scientists can observe their replication and effects. And this is how we learn what makes a virus infectious and how it damages the cells and how to test antiviral drugs. And these all things happens always inside a high safety labs and under strict ethical rules. So now let's go to the methods of the viral cultivation. So these are the three methods used. First one is animal inoculation, then embryonated egg culture, then tissue culture or cell culture. Under it comes organ culture, explant culture and cell line culture, which is the most commonly method used. So let's go into the animal inoculation. So in the animal inoculation, it is one of the earliest methods used. Before, Reed and their colleagues, they used human volunteers for the yellow fever virus. And then it got replaced due to the ethical and safety concern. And now the commonly used animals are mice, guinea pigs, rats, hamsters, rabbits and all. So you can see in the diagram how it is inoculated into the animals. So let's go deeply into it. Like we'll see what are the routes of inoculation. So the routes of inoculation in animals, there are four main routes. One is intracerebral, then subcutaneous, then intraperitoneal, then intranasal. You can see in the diagram how it is inoculated. And after the inoculation, the viruses multiplies and the symptoms appear. And after the appearance of the symptoms, the virus is isolated from the tissues. Even the mice can be sacrificed and the tissue histology is studied. So it is mainly used for research purposes and for some viruses which are very hard to cultivate, like arbovirus and Coxsackie virus, etc. So the advantages are to study the pathogenesis, immune response, antibody production, and the disadvantages are they are very expensive and the difficulty to maintain the animals and ethical issues related to it. Second method, that is the inoculation into embryonated egg. So this method was first used by Good Pasteur in 1931. An embryonated egg of chick or duck can be used. So here we are looking the example of 7 to 12-day-old embryonated hen's egg. Okay, first the surface is sterilized, then the virus is injected, and then it is sealed and it is incubated at 36 degrees for 2 to 3 days. And after the incubation period, this egg is observed, that is, the virus can be detected by the death or the lesions on the embryo. So we can use the duck egg since it is much bigger, so it can be used for the better yield of vaccines, especially for the rabies vaccines. It is for the preparation of inactivated non-neural vaccines and all. So now let's see the routes of inoculation into the embryonated egg. So these are the routes or the sites where virus is inoculated into the embryonic egg. So the first one is chorioallantoic membrane. So you can see it is a membrane, that is why it is preferred for the pox viruses like variola, vaccinae viruses and also herpes simplex viruses. And so we want to see the pox or the lesions, it is visible in that membrane. So next one is allantoic cavity. So it is a little bigger cavity, so we can use it for getting better yield for the virus vaccines for yellow fever, rabies and all. And then comes the amniotic sac. So it can be used for the primary isolation for influenza virus and mumps virus. Then it is yolk sac. It can be used for the rickettsia, chlamydia and some viruses. So the rickettsia and chlamydia, these two are bacterial. And since they also act like They cannot be grown on the artificial medias, so they are also cultivated in this form. So now let's go to the tissue culture. And in tissue culture, it is the growth of tissues or organ fragments in a nutrient media for the virus's propagation. So the three methods or three types are organ culture, explant culture and cell culture. So in the organ culture, it is previously used for some fastidious viruses that have specific affinity for specific organs. For example, the tracheal ring for the coronavirus. Okay, so next one is the explant culture. So in explant culture, taking a small piece of tissue, that is which is called as an explant, from an animal and putting it into a nutrient media to let the cells grow outside the body. And now then we inoculate virus into it. Now it's rarely used. Okay, so the most common method which we are going to discuss is the cell culture. So the cell culture, the cells are grown in vitro in glass or plastic petri dishes with nutrient medium, and the medium contains amino acids, sugars, proteins, serum, buffer, antibiotics and phenol reds, whatever things which is required for their growth. After the inoculation, the cells divide, there will be a monolayer formed and we can see the viral growth. Okay, now let's go into the types. The first type is primary cell culture. You can see it is a normal cells freshly taken from an animal, and these are able to grow only for a limited time and cannot be maintained in serial culture. So thus it can only be used for the primary isolation of viruses and for the vaccine production. So the examples are monkey kidney cell culture, chick embryo cell culture. Okay, now the second one is semi-continuous culture. So they are diploid and they contain the same number of chromosomes as parent cells. So they can be subcultured up to 50 times by serial transfer. Okay, and it can be used for the isolation of some fastidious viruses and production of viral vaccines. And the examples are human embryonic lung strain, rhesus embryonic cell strain. Okay, now the third one is continuous cell line. Okay, so they are derived from the cancer cells, and it can be maintained either by serial subculture or by freezing it in the deep freeze at minus 70 degrees Celsius. And due to the derivation from the cancer cells, they cannot be used for the vaccine production. And the example is HeLa, which is Henrietta Lacks. So let's come into a conclusion. So I told you that viruses need living host system for the growth. And the three main cultivation systems are by using inoculation into the animals, inoculation to embryonated eggs, and tissue and cell culture methods. So by these, we can cultivate the viruses. Thank you.